On the luminal surface of proximal tubular cell, sodium-hydrogen exchanger. On the luminal surface of the intercalated cell, potassium-hydrogen ATPs and hydrogen ATPs. On the basolateral surface of the proximal tubular cell, sodium bicarbonate co-transporter and the anion exchanger protein. On the basolateral surface of the intercalated cell, only the anion exchanger protein. Into the proximal tubular cell, carbon dioxide diffuses from the interstitium and with the help of the carbonic anhydrase enzyme, reacts with water to produce carbonic acid, which dissociates to produce the hydrogen ion and bicarbonate ion. Into the intercalated cell, carbon dioxide diffuses from the interstitium and with the help of the carbonic anhydrase enzyme, reacts with water to produce carbonic acid, which dissociates to produce the hydrogen ion and bicarbonate ion. In the proximal tubular cell, hydrogen ion luminal secretion is coupled with sodium ion intake as sodium ion is moving from the tubular fluid into the cell down the concentration gradient, and therefore hydrogen ion is said to be secreted into the lumen via the secondary active transport. In the intercalated cell, however, there is the primary use of the ATP molecule by the potassium-hydrogen ATPs, causing the pumping of potassium ion into the cell while hydrogen ion is secreted into the lumen against the concentration gradient, and that is why it is regarded as a form of primary active transport. The active secretion of luminal hydrogen ion by the hydrogen ATPs is also a form of primary active transport due to the direct use of ATP molecule and the pumping of hydrogen ion into the lumen against the concentration gradient. In the proximal tubular cell, movement of 3 bicarbonate ions into the interstitium is coupled with movement of 1 sodium ion along the same direction thanks to the help of the sodium bicarbonate co-transporter. Movement of 1 bicarbonate ion into the interstitium is also coupled with the intake of 1 chloride ion via the anion exchanger protein. In the intercalated cell, movement of bicarbonate ion into the interstitium is coupled only with the intake of chloride ion by the cell due to the presence of only the anion exchanger protein along the basolateral surface. In the lumen of the proximal convoluted tubule, secreted hydrogen ion reacts with filtered bicarbonate ion, producing a carbonic acid which dissociates to produce water and carbon dioxide. Carbon dioxide diffuses into the proximal tubular cell. In the lumen of the collecting duct, secreted hydrogen ion reacts with phosphate, producing the H2PO4-, which reacts with the sodium ion to be eliminated in the form of NaH2PO4.